All right, here we go. So it's my pleasure today to introduce Amy Arnsten, who we have chatting with us today. Amy's a professor of neuroscience, I guess neuroscience and neurobiology at Yale. And I've been a fan of Amy's work for a very long time. And I can actually pinpoint the first paper of yours that I ever read and what I thought about it. So when I did my PhD, I was sort of coming at the brain very much more thinking about sort of behavior, neurodegenerative diseases, and sort of thinking about function and anatomy at a kind of macro scale. But when I went to do my postdoc, which was with Trevor Robbins in Cambridge, I wanted to understand the neuromodulatory systems much more seriously, and in particular noradrenaline. And I remember reading your paper, sort of near the beginning of my postdoc, the Through the Looking Glass paper, which I think is from 2000 now. And it's a lovely paper, especially because it has this beautiful Alice in Wonderland illustration and all the content in it is great as well. But I remember reading that paper and just sort of seeing this kind of developing this line of thought that went all the way down from the machinery of kind of molecular biology through sort of, what the cells are doing and what neuromodulators are doing to sort of large scale organization of these neuromodulatory systems to then sort of making serious conclusions about behavior and how we interact with the environment. And I'd never read a paper like that. I'd never read a paper that was kind of pulling that whole spectrum in together. And I fell in love with that type of thinking. And I was obviously exposed to a lot more of it at Cambridge. And when I got to know more of your work, I realized that probably that line of thinking of really thinking of that whole spectrum probably was coming from your work with Patricia Goldman-Rakish, who was sort of the first one kind of pulling these things of, the weeny details of molecular biology, all the, through sort of long range connections and all the way to behavior. And I guess, I just wanted to sort of introduce you as someone who, had a big influence on my thinking, particularly as someone who is not only sort of thinking across these scales, but sort of taking them really seriously and sort of working across them, which is, I think really amazing and how I've tried to keep thinking in my career. Not always getting it right though.
That's so moving to hear. And you are sure right about Cambridge being such an important place for this that I did a brief postdoc there with Sue Iverson,
Mm,
who was also trying to really capture how neuromodulation altered behavior. And then Trevor, as you mentioned, and Angela Roberts, I
yeah.
think they have totally changed the landscape and really giving us this understanding of how molecule circuits really are relevant to, especially like with Angela's work now on depression, Trevor's work on so many different disorders. So you certainly trained in a fabulous place and a beautiful place too.
Yeah, yeah. And I suppose, I mean, I think both, and Mac is a big fan of your work as well. So we have, I think, lots of things that we could talk to you about. But I wondered if, a kind of good opening gambit to kind of start big before we get to more of the details is to actually maybe to get your thoughts on this sort of importance of kind of thinking of across scales and how we can achieve it. And maybe in particular, because a lot of our work, has been in sort of human neuroimaging. And as we were just chatting before we started, that neuroimages don't often make contact with that level. And I just wondered if maybe that's a good place to start the chat.
Yes, I'm old enough to have been around during the origins of brain imaging, the origins of molecular neurobiology and systems neurobiology, when they were all small enough fields that they all really talked and interacted. And with this great ballooning, which is wonderful in many ways, of so much research, I've watched the entropy just pull these apart. And now huge fields where very often people don't have the training to even be able to speak to others. It's intimidating jargon to hear the molecular and, oh, I can't do that. I don't do that. And yet we really need to go across that whole range to be able to understand how genetic insults lead to symptoms, how to design treatments that really work. And so it is our task, and it's important to not feel afraid and to be curious and just dive in.
Yeah, I couldn't agree more, Amy. And I think from our perspective, or at least, from my experience, I think this notion that we have to kind of do it all on our own is kind of misguided and naive and kind of like stops the conversation. And for me, the biggest thing I've learned is if you don't understand what a term means, go and meet with a collaborator or, have a coffee with the local molecular biologist and just ask them, when you're doing an RNA-seq, why is that different from merfish? And like, why should I care about the differences and what kinds of conclusions can I make? And I had a background in biochem and then I went to medical school. And so I learned a lot of this kind of cellular level detail in physiology and then got into neuroimaging and it was just sort of all gone. It was sort of like sort of wisps in the air. And all of a sudden now I had all these bold patterns and default mode networks and functional connectivity, these awful terms that don't really mean what they're trying to say. And you have all these terms, but you don't know how to find the way to kind of connect them down and link them up with the kind of way the system's built. And so for me, it's really been about kind of meeting with people and trying to, break down that entropy and kind of create overlaps. I'd actually really I'd love to know what kind of how you got started on this journey. I think you were kind of a real pioneer in this space and thinking and doing the kind of really hard work when it wasn't a common thing to be doing. I mean, how did you find your way into that? Was a curiosity? Did you have a road to Damascus type of a thing where you're like, oh my God, it has to be like this? Or was it more, wow, that's a beautiful circuit and I want to understand it better and peel back the layers? How did it all come about?
So, first of all, you have to know I'm old enough that I was really getting in on the ground floor of a lot. In some ways, that was a more fun pioneering time because everything was wide open, um, although not wide open for women. So, and especially women who wanted to have kids. So, lots of hurdles, but a very different kind of feeling of a very broad landscape in front of us. And I had been seeing while a teenager difference, just heartbreaking differences in how mental illness was viewed with utter ignorance. And I was able to compare it to my father had a heart attack, and a little bit later, his sister had what they called a nervous breakdown, and able to see how knowledgeable even then the cardiology community was, how supportive everybody was. Everybody knew what to do, saved him. There was so much support and sympathy versus the total ignorance around what was happening in my aunt's brain, how to help her, the stigma. So I realized even as a teenager, we really needed to understand what changes in brain in mental disorders. So that was really what pushed me into neuroscience. Also volunteering in a state psychiatric hospital to learn more about the clinical side of things. And and then looking for clues, science is very much a treasure hunt, right? And one of my first clues, like with Claire, was norepinephrine, or to say it in the Cambridge manner, noradrenaline. And just seeing that it went to all these fascinating places in brain. So, it could likely have a large influence in these big state changes, which we were correct about. And then in grad school, I happened to learn about prefrontal cortex, and that really was an exploding star because I got to when I learned what it did, it really matched with what I understood about the symptoms of mental disorders. Plus, it's so fascinating. It is our highest order cognition, etc. So I have studied it ever since. And so I was going to Cambridge to do a postdoc with Sue Iverson on dopamine. But when I heard this woman, Pat Goldman Rakesh, gave a little seminar at UCSD there, where I was getting my PhD, and there were like 10 people in the room. No one had heard of her. And when I heard her work, I knew I had to work with her. And so that changed everything. Yeah. And so
What were her
watch,
talks like? I'd love to know because I've read a lot of her papers but never had the chance to meet her, and it just the kind of wide sweeping neuroanatomical detail is just like mind blowing. But also the ability to communicate it on 2D was it's kind
yes.
of I just love it. Sometimes she'll flip a brain around and you're like, oh yeah, of course, that's such a great way to look at it. And it was just like this brilliant mind. Yeah, I'd love to hear.
Yes, having that artistic ability, incredible surgeon, piano player, so many skills that enhanced her scientific ability. At that point in time, she was still mostly doing neuroanatomy, including with development, but also had already discovered or recently discovered how important dopamine was to prefrontal cortex. And from her data and in the footnote, I was able to see, well, it's not just dopamine, it's norepinephrine too. But that came later. Um, so what I got to see as she was evolving her lab at Yale, and I was still a postdoc, is how uh, she she would ask the most important questions, and she didn't let the whatever methodology she had limit her. If the question required a different kind of method, she would collaborate or very often bring that expertise into her lab. And so she was one of the first to be doing this multidisciplinary work. At the time that I was a postdoc, almost all other labs would specialize in just one arena and they would stick in their lane. And the technology was really um, very much the limitation of the questions they could ask. She, having an amazing prefrontal cortex, was very top down and said, no, the question is what guides what we do. And things that were so ambitious that mortal beings would be like, oh gosh, how on earth are you going to get at that? She would just go ahead and do. And she'd often
Like a
have,
bravery, yeah, yeah,
oh, yeah, not, and also the skill to do it.
yeah, yeah.
Really doing magnificent work that really came out and addressed major questions like the neurobiology of schizophrenia, providing a cellular basis for symptoms that people before that would have thought were really not answerable by science. And the same attitude had been about prefrontal. Oh, that's not an area that we can address. That's a mushy area. It's
Yeah.
not something we can address with science. So she just kept not listening to what people said of you can't do this. And I think that's an advantage of being a woman at a time when women weren't allowed to be science is like, okay, I'm not allowed to do this, and you're telling me I can't do that, so I'm just going to do what I want.
Just thumb
When
the
I think,
nose, yeah.
yeah.
I
So,
love that.
and
Can I ask you a question along those lines actually? I don't know if you've ever played around with NeuroTree before, this kind of lovely online sort of corpus of who trained with who. And I was
mhm,
I was playing around one rainy day and I was sort of looking up some of the people in science that I really admire and I found Pat's sort of links and she was trained by Wally Norter. I was,
mhm,
an anatomist back in the day. But I didn't know that he also trained Anne Graybill and Susan Haber, who are sort of three of the most prominent female neuroanatomists in our field. And I just
yeah.
thought, what a great example of championing young women in science back when it was really not common. And so I just wanted to know if you had any experience with that or if you know more information, because all I have is these three links on a on an old website.
Neurotrain. There was a symposium to honor him when he was retiring, and Pat spoke, and she gave, of course, millions of seminars, and usually was preparing them at the last minute. This one she cared about so much, and she gave one of the most magnificent talks I've ever heard her give. And it really came from loving him, honoring him, being so grateful to him. Yes. And how the methods he taught her gave her her talisman. So She was actually at NIH, and then that was a sabbatical where she was learning those methods. And I met Pashko because of being in Boston. What she discovered with him was that there were, when they looked at connectivity in prefrontal, that there were columns in prefrontal, just as there were ocular dominance columns in V1. I mean, different size, shape, whatever, but nonetheless, a columnar organization there. And that was her talisman, that you could use the same tools to solve prefrontal as you could with primary visual cortex. And she kept this image of the columns in her office her whole time. And when she died, I went to find it, and because it used to be on the wall, and I was, oh no, it's not there anymore. And then I found it under her desk, right where she put her feet. So it was always there. You know, a true talisman in that way.
That's incredible. I'm so happy to hear that that was a sort of wonderful, enriching experience because you kind of, you sort of see these links and you quite never quite know what the situation was like. So that's amazing. Cool. Yeah, Claire, I don't know if you wanted to ask someone.
Yeah, gosh. So many, so many places to go. I was thinking a bit when you said at the beginning that, sort of the reason that you got into neuroscience was thinking about mental illness and sort of how poorly it was understood and treated. And when I think about kind of the sort of basic science to clinical realization of guanfacine, I mean, you must pinch yourself to think that I mean, I know it's a big team and it was sort of on the back of other work, but to think that, you started out in this field sort of, thinking and wondering about mental illness. And then one of the end products has actually been to sort of find a drug that really helps with these conditions and possibly with other conditions. And I don't know, I just think to sort of see that kind of going from basic science all the way to the clinic is incredible. And I always have this thing in mind, again, when I was trying to understand, trying to understand more about noradrenaline and the receptors and things. And I remember, I think it was the first experiments that you and Pat Goldman Rikish did, giving the older monkeys clonidine, and that they were described as, very sleepy, but they could perform really well. And that's, for some reason, that's kind of when I try to think about, alpha 2 receptors and presynaptic and, autoinhibitory receptors. That's how it kind of all makes sense in my mind, thinking about, clonidine sort of dampening things at the level of the locus coeruleus and the sort of, imidazolin receptors and sympathetic outflow, but then having these, postsynaptic effects at the DLPFC. And I don't know, that just must have, to sort of see that thread all the way through in your career must be such a fascinating thing and such a satisfying thing.
It has been when things are hard, and I'll tell you they are sure hard right now in the U.S. Given the chaos caused at NIH. It's a lifeline to hold on to. It is worth it because it's really helping people. And I get amazing emails still from people saying how guanfacine is saving them or their kids in life-changing ways. And I'm so grateful to hear it because usually you don't get to hear what's happening. The sedation, to come back to, we think it's also due to alpha 2B actions in the thalamus. And part of why we think that is dexmedetomidine is preferring alpha 2B, and it's so sedating, it's used as an anesthetic. And alpha 2B in thalamus is only in primates. It's not in mice. So the mouse models didn't reveal this because they don't have it there. But when you look at for example, human, you see very dense alpha 2B in thalamus.
Okay,
So
but Dex still works in rodents too, right? So it must have effects on other receptors that just aren't as potent. Is that the idea?
yes, so the alpha 2A that we've been talking
Yeah,
about,
yeah, yeah.
so with the yes, quieting the locus coeruleus as
Mm.
well, yes.
That's
Um,
fascinating. Yeah, this cross-species difference is question that looms so large. I think I've been interested in, many different parts of the brain, but one in particular of late has been the cerebellum and thinking about massive changes in outflow, the
Mhm.
sort of size of the cerebellum, but also the outflow of the cerebellum to the thalamus. And you go and look at all these incredible new technologies in mice. The Allen's really, driven this huge catalyst of new methods and new techniques. But you kind of fundamentally can't ask the question that you want to ask about something that we can do in humans. Maybe it's due to the circuit with the same technologies because that animal just doesn't have that elaboration. And it's such a you feel like you've got one arm tied behind your back and you've got half your eyes closed or something like that. And that must have been kind of how you felt trying to make this progress throughout your career and seeing things. I'd love to hear kind of how you don't let that slow you down. How do you kind of fight through that inertia of not having the tools that you really need to kind of ask the questions that you want at scale? Do you have any, advice or thoughts on how to navigate that?
Well, the most important thing is you start with the human and the symptoms or the genetic insult, whatever, and you anchor yourself there. That's the fundamental truth you're trying to get at, and you're always then with your animal work saying, is this indeed relevant to that, or am I going off on a tangent where this animal has a very different nervous system and it's taking me left, whereas in the human it would go to the right. Because there are things that are opposite. You get to see, for example, that with neuroinflammation, mice are built to withstand nuclear war. They're just so resilient. They have so little neuroinflammation compared to humans, or, we're like, oh my gosh, there's something wrong. Shut down prefrontal. So, when we were trying to make these, mouse models to look at some inflammatory mechanisms. And they're like, oh, yeah, we can handle that. Just resilience here, resilience there. So it was very frustrating that we couldn't get the same phenotype that we knew was going on in humans. And so it's really important that whatever model you're using, you make sure it really is relevant. And then sometimes you can just capture one aspect of it and model that, whether it's in a dish or in an animal, but always making sure that it comes back to the human. And for dorsolateral prefrontal and certain other higher circuits, you can only do that work in a primate and an advanced primate at that because those mice, rats just don't have those parts of the brain. And yet, I like to say that dorsolateral prefrontal cortex it's our hero Achilles, but it's also our Achilles heel. So it has all this built-in mechanisms to go offline with sickness, with stress, with inflammation related to the sickness. So when you have these very powerful mechanisms to actually shut off prefrontal within moments, very rapidly, and to actually erode connections with chronic stress. Um, I think that's why prefrontal dysfunction is so common to so many mental disorders.
Yeah, I guess, sort of on the theme of, yeah, this kind of, challenge or at least thing to consider of cross-species differences, even just the idea of kind of the differences in sort of morphology and neurotransmission, across the cortical hierarchy, which you've, shown in so many cool ways. And when I was sort of reading some papers yesterday in preparation for this, and I sort of, even though we sort of just take it for granted but stopping to think about just how amazing it is, this sort of architecture, at least that you've shown in the prefrontal cortex, that can sort of flexibly change in such moments, yet it doesn't lay down LTP, it doesn't sort of, commit this sort of, long-term memory, but it's sort of changing the network configuration sort of so briefly in a way that it can just sort of, in a fleeting way, sort of come and go. And I was thinking more about your paper sort of showing that, it's the sort of, the elongation of the spines and obviously the neurotransmission, all this kind of concert of neurotransmission and sort of morphology that sort of makes parts of the brain special, that they can have this kind of fleeting engagement and flexibility. And I don't know, I think it's something we don't sort of stop to think about much, and sort of how that plays out in terms of, real genuine differences in morphology and neurotransmission that then sort of permit these, very different kind of cognitive or behavioral trajectories.
Yes, how we make something so dynamic. And when if you think of it, working memory, abstract thought, it's very ephemeral, our mental sketch pad. And it also has to be really responsive to the environment. Oh my gosh, look what's happening, something dangerous. And so it makes sense that it would have different circuitry and different molecular regulation than, for example, hippocampus, who's trying to lay down a memory for a very long time. It's almost an opposite job to do. And one of the things about the morphology and the molecular, how they interact, is just beautiful to me. And seeing actually in primate at least, most of the spines and cortex are long and thin, like 60 to 70%, certainly in layer 3 of dorsolateral prefrontal. And it turns out that thin structure, especially a thin neck, but also having not too large a spine head, is really critical for this dynamic nature, because what's allowing these connections to either be strengthened or gated out completely so that you take prefrontal offline with a threat, for example, is you build up very rapidly from the release of high levels of catecholamines, high levels of cyclic AMP, PKA, and calcium. And these open a whole series of potassium channels that are concentrated on the spine neck and relatively close to the synapse sometimes. And when they open, it just gates out. It's like, okay, you can no longer talk to this cell. We are shut down. You are quiet. And that can happen like that, right? Because danger can happen like that. And then having all these regulatory mechanisms to chew that up or stop it from happening, that allows prefrontal connections to reconnect. It's all right now, we're safe. We can think again. And one of the things that's so rotten about inflammation is it destroys those very mechanisms that allow us to chew up the stress response and keep it under control. And so then we have this chronic weakening, and you actually lose the connections. And that's when architecture really exaggerates what's been happening chemically. So you get loss of prefrontal connections and actually more connections in the amygdala and primitive circuits. So now, I should back up and say the whole point of what's happening, and norepinephrine is key to this. It's such an orchestrator of brain state. High levels of norepinephrine engaging low affinity alpha 1 and beta 1 receptors, rapidly taking prefrontal offline, but at the same time strengthening amygdala, and amygdala then strengthening striatum. And also strengthening sensory cortices. So you strengthen these primitive, reactive, habitual circuits, the sensory motor circuits, while taking prefrontal offline, and that seems to get exaggerated with chronic stress.
And then, so to then link this back to the guanfacine earlier, so it's an alpha 2 agonist, and normally we think of that as an inhibitor because it's a GI/O, which means it's going to come in and downregulate cyclic AMP, and then you're going to get other kinds of internal effects. But you spoke at this conference that I was at a number of years ago when I was just a really young postdoc, about the fact that that can actually have internal effects that can actually help the cell become active. So it's not just that you're stopping the efflux of potassium, but you're also changing things like HCN channels and all these other complicated internal dynamics that are keeping the sort of cell active in this really interesting regimes where it can still process information. And I think that's such a lovely counterintuitive, like so deeply fascinatingly biological way to think about what we need to actually do to support higher cognition. I don't know if you want to say a bit more about that or talk about yeah, anything. I just find this whole thing so fascinating.
It brings us back to Alice in Wonderland, and that everything is through the looking glass,
Yeah,
and that has opposite needs to hippocampus and sensory motor cortex. That cyclic AMP actually harms it at these higher levels because it's opening. So HCN channels that are partnering with Slack potassium channels, then PKA, KCNQ2, and the calcium, because it causes calcium release from the ER and increases calcium entry through voltage gated calcium channels. And the calcium opens SK potassium channels. So you have this, you can tell Mother Nature is like, we are going to make sure you
yeah.
disconnect. They're doing this in at least three different ways, and I bet there's a bunch of others as well, because it has such survival value under some conditions. Not if you're solving climate change, but if you're cut off on the highway or there's a saber-toothed tiger or something like that. But yes, so this idea that of having opposite chemical needs to sensory motor, amygdala, hippocampus. And I think that's kind of beautiful, but I have had a long history of people telling me, no, I'm wrong. It's opposite. No, cyclic AMP is a good thing. You're doing this wrong. So I very often get that still. But the alpha 2A receptor, where guanfacine acts, has very high affinity for norepinephrine.
Mm.
So it's getting engaged under conditions of safety, when there's something interesting and we get a little burst
It's
of
a little
norepinephrine.
bit, yeah, yeah.
Yeah, and so it seems to be a safe signal, not only in prefrontal, but throughout brain and the whole nervous system, that you get to see this play. For example, alpha 2A is anti-inflammatory, whereas alpha 1 beta
Mm.
often pro-inflammatory on glia, on macrophages. So throughout the whole neuroaxis, alpha 2A seems to be, hey, we're safe, it's okay, you can be a philosopher now. And and so I think that's why guanfacine's now being used not only for cognitive disorders, but also as an anti-inflammatory. So there's studies in progress, and one I think completed, showing it helps with delirium, which makes sense, right? So here you have prefrontal disconnected either due to anesthesia or especially infection, and that this is helping to reconnect prefrontal to allow cognition to return.
I also think this is such a lovely kind of synthesis, and I think it actually fits really nicely with some recent work that's been coming out as people have been able to actually record directly from not just locus coeruleus neurons, which has been this incredibly difficult imaging problem for so long, right? It's this tiny structure in the
Yeah.
pons, and it's right next to a whole bunch of areas that you really don't want to damage because otherwise the animal doesn't particularly stay alive for very long. So it's been a really hard problem. But just recently, a couple of groups have cracked it, and Vincent Breton Provencher works up in Canada. Did a really beautiful study that was in Nature where they tracked in an oddball paradigm. They tracked the fibers of the LC that were projecting up into the cerebral cortex, but also LC neurons. And they actually showed something that I think is really consistent with the architecture you just suggested, which is that during the early parts of the task, what you find is that a few different LC neurons will spike, kind of a little bit like in a cacophony, almost like there's a murmuring of discussion amongst a crowd. But then the moment that there's an oddball, all of a sudden there's this huge jump in all of them. They kind of light up together en masse. And so you can imagine that if you were in this sort of philosopher zone, intermediate amounts of noradrenaline are being released, you're going to kind of recruit this a little bit over here, a little bit over there. You're getting a little squirt of noradrenaline. But then all of a sudden something bad happens, the saber-toothed tiger, bang, the whole circuit goes network reset, Susan Sarah style, and now you can change your behavior. And I love the idea that the organization here of this behavioral function is not sitting in the DLPFC, it's not sitting in the LC, it's the way that that whole system works in the context of an animal embedded in an environment. And I love the idea of biology finding this solution to this problem, but not, localizing it. And I think this kind of then brings me back to kind of the frustration. I think, Claire and I share this frustration a lot of the people in the field do in imaging, because for a long time we've gone, hey, look, I found, working memory. Here it is. It's sitting in this spot. And I think what we want to say is actually there's this big complicated brain and allows the animal to do working memory in this context. And let's not be too quick to say it's right there. It's in the system. And trying to come up with the kinds of ways that the answers should look so that they're satisfying, I think is a really important step for us to take as a field. I'd love to hear your thoughts on that and whether you think that we're making progress or we're kind of dooming ourselves to further and further failure as we move forward.
So two things to go back to locus ceruleus. So Gary Aston Jones, Steve Foote had done a lot of early work that really actually showing that during that oddball you're getting locus ceruleus firing, that you get a P300 in the EEG, and that depends on having locus ceruleus firing. And then Gary and others showing also that difference in the phasic firing to the oddball to under a stressful condition, it's tonic firing. So you're just having constant firing. And so you can imagine it's in that latter situation where now you're engaging these low affinity alpha 1 and beta.
Absolutely. Yeah, Vincent interpreted his results in light of Gary and John Cohen's adaptive gain theory. And I think, yeah, I should have definitely mentioned that. My apologies.
Just going, yeah, when you're old, you remember who did things first because they were your friends. But and then the human imaging work that really fits with that, a lot of it out of the Netherlands, showing that when you stress someone, you have this reconfiguration of functional connectivity where you have then weaker dorsolateral prefrontal and stronger anterior cingulate, and this, like connections of anterior cingulate with amygdala, etc. What they then called salience network, and I think they were the first time to call it that, and that they could stop that happen by giving a beta blocker. So to bring it back to the norepinephrine, and this idea that norepinephrine is having such a key role in orchestrating who's able to talk to who, and the kind of different roles the same region might play depending on who it's talking to, as you're saying, in getting at that it's not just this one spot. On the other hand, what you also get to see, though, from lesion studies, including really old studies where humans with lesions back in the 1950s, there were a lot of lesion studies in monkeys, for example. You get to see what's necessary. And so even a small lesion in prefrontal can really impair working memory. And Pat did a study showing this was a temporary lesion, that you can see working memory related neuronal firing in both dorsolateral and parietal, as a monkey does a working memory task. If you cool prefrontal, you lose most of that activity in parietal. But if you cool parietal, you don't lose that much in prefrontal. So we get to see a lot of reflected activity with neuronal recordings as well as with functional imaging. But it's usually only these temporary lesions are particularly illuminating, although in humans, even more moving when it's a real lesion. You get to see the unique contributions of different areas. So they really do bring something special to the table often.
Yeah, no, I agree for sure with the lesion studies, and even sort of way back in our PhD, Mac and I both started working in Parkinson's, and myself in Parkinson's and frontotemporal dementia, and these are multi-system disorders, so they're not anything like a neat lesion study, but it really does let you understand the brain in a different way. Sort of seeing how it unravels is gives you such an insight into sort of how to think about it.
Yes, and especially as I think with Alzheimer's, as you watch the pathology kind of march through the circuits
Yeah.
and the progression of symptoms that arise as the pathology occurs, which is both heartbreaking, but also it fits startlingly well with what we know about the function of those cortical areas.
Yeah, there's some really lovely work in the last few years from uh, some recently from David Weinschenker's lab, where they're looking at trying to sort of characterize this prodromal phase of Alzheimer's that they think might be sort of
Yep.
neurogenergic state, which I mean fits so well with these kind of symptoms that we see before Alzheimer's. Yeah, so that's another lovely thing to think about. But one thing I was thinking about asking you, I mean, I really love the neurogenetic system, and I also, I think dopamine gets so much attention. It always steals the limelight. But whenever you do anything, and rightly so, when you do a study or write something that's focused on noradrenaline, you always need to kind of add the sort of caveat in that these systems work together in this neuromodulatory cocktail, and they're all and even with drugs, it's kind of, I think of it like a Rubik's cube. You can't sort of change one side of it without affecting the others. And I think sometimes we ignore that, but I guess we also ignore it because it's really hard to not only to think about them all together, but sort of experimentally to sort of look at them together is technically really challenging. And something I really love about your work is, in the thinking about the sort of inverted U and the role in working memory, of kind of nailing down almost the relationship between dopamine D1 receptors and noradrenergic, being able to kind of at least take two of them and in one sort of area and in one behavior, but actually really understand how they're sort of synergizing. And I don't know if you sort of have any thoughts on that. I mean, for me, it's obviously what we need to be doing, but it's hard as well, isn't it?
Yeah, no, I need about 18 lifetimes to realize, especially if we're going to ever take on serotonin, we need 17 lifetimes. But norepinephrine actually drives the dopamine cells, so it's really important there. So we have seen that not only no norepinephrine and dopamine, but acetylcholine are critical to prefrontal function. This idea that the arousal state has such powerful effects on these higher cortical circuits. And we're guessing that it's not just dorsolateral. Speaking of needing 18 more lifetimes, what does it do to orbital, to dorsal medial, prefrontal, to parietal, et cetera? That we've learned that acetylcholine is essential to NMDA neurotransmission in dorsolateral prefrontal, that it plays the role that AMPA receptors normally do. And since acetylcholine is only released when we're awake, not in deep sleep, this idea that we may be unconscious in deep sleep just because there's no acetylcholine. And then in REM sleep, we have acetylcholine, but we have absolutely no norepinephrine or serotonin. So is the difference between dreaming and waking just having norepinephrine and serotonin on board? It's a very reductionist and silly thing to say, but it's what dramatic changes in our mental state, right? So and seeing how critical acetylcholine is to neurotransmission in prefrontal, and then this really needing a little dopamine D1, but then too much is what you get with stress exposure. And I think that's part of what makes dopamine therapeutics so difficult? It's all happening at the same receptor, this narrow inverted U with D1, whereas norepinephrine has had the kindness to separate its good from its bad receptors. And so we've made a lot of progress with therapeutics with beta and alpha 1 antagonists, alpha 2A agonists. Um, so all of those can treat stress disorders, and the alpha 2A, even non-stress cognitive disorders. And I think it's because of this heterogeneity in affinity for norepinephrine and having this range of receptors, whereas with dopamine, we're mostly stuck with D1.
Yeah. I Something I think about with the like you said, the narrow inverted U, and I don't know if it's a silly question, but when the inverted U is so narrow, and so sort of it's so specific to particular tasks and things like that. And if you were, if you were designing a biological system, you would think, let's make it wide so it's really working well, for across the board. But it has this, I don't know, is that something you think about? I mean, in terms of kind of, obviously the narrowness is a vulnerability, but I'm sure it buys you, so much in terms of, the system existing almost on a knife edge, that it can be so flexible and changeable.
Yes, no, it seems like, and given we see the little bit of prefrontal that mice have going offline as well with stress, that this really has had survival value. That switching from prefrontal thoughtful, mouse as philosopher, to this more instinctive, reactive, habitual state under threat has saved lives enough that it's really been what's carried through across evolution. And I think it's still true with humans in a lot of life-threatening situations, even in modern life. I'm always using the cutoff on the highway because that's something that most of us have experienced. And I'm just so aware. There I am thinking, how am I going to write this grant? How am I going to respond to this reviewer? And then suddenly here's this truck in front of me, and I very much stop thinking, or at least maybe swearing, but I'm not thinking anymore. And you just slam on the brakes, and that your striatum has been trained over years and years to do that. So you could do it without thinking. That's what boot camp is about for the military, about all sorts of drills for firemen and policemen, so that those people who are working under threat all the time can have a striatum that knows just what to do, even if their prefrontal is offline. And so you can see where that saves your life, and then you can go on and have kids, and those genes get passed on for keeping prefrontal offline with threat. But it's so hard when you're needing to be able to think about complexities, to be able to plan ahead. And so much of what we're seeing right now in the US is how disastrous it is when people making decisions aren't using prefrontal cortex, making short-term selfish decisions, and how much everybody then pays.
Yeah, so like the way it seems to me from afar, as someone who spent a lot of time in the US, I'm actually a dual citizen of the United States and Australia, and I'm heartbroken about what's going on. But it seems a lot of what's going on is people leveraging the habitual response to sort of actions, getting people really emotionally worked up about a particular outcome and maybe overblowing its probability. And it's not just happened in the United States, it's happened all over the world. I think we haven't really learned how to use this kind of more distributed access to information that's not as curated, it's not as well crafted, it's not as well fact checked, and a lot of it kind of touches us back into this notion of kind of almost like we're back in high school and there's just the gossip is what matters. Who said what? Oh, I believe this is the truth. It's like there's a reality out there and there's actually something happening, and these things either work or they don't. And we have evidence bases that are behind a lot of the decisions we make for these really, really hard, counterintuitive situations. Like how does it make sense that biology has learned this little trick to turn on your cells and circuits when you're a little bit stressed, but if you're too stressed, turn it off. This is a really counterintuitive thing to think about, but when you actually look at the details, they're there. And I think there's a really important thing for us to grapple with if we're going to survive very long as a species, is to like, how do we start to get back trust in this idea of learning and evidence-based information that's not just about opinion and misguided kind of incentives. Anyway, we could rant
Yeah.
for hours, Amy.
I think
Well,
we,
I can, just if I can bring that back to prefrontal
yeah,
cortex, that's the function of the frontal pole, is to have insight
Yep,
about oneself and others, and being able to evaluate information skeptically and
yep,
synthetically. And I'm very struck from, for example, the work out of the Yale Stress Center showing that with chronic stress in humans, that's where you get the most atrophy, is in that frontal pole. And I think of, yes, the kind of scare tactics, also the kinds of adversity a lot of people have had to live through, still living through. And then you add COVID, which also erodes that same gray matter, that people are often in a worse actual biological state to be able, they're in a vulnerable state and being taken advantage of.
I was just going to suggest we could sneak guanfacine into the drinking water.
Yes.
I'd be for that.
I love it how we've gone from like this beautiful talk about brains and now we're doing,
Yeah, this is
into
how
the
we can solve
reservoir.
the problems in the universe.
Instead of fluoride, we'll just
Exactly,
get
yeah.
that. Yes.
Yeah, the teeth seems like a problem that's less important to solve at the moment. Uh, um, Amy, I realize we're coming up to the hour, and I don't want to take too much more of your time. Is along with what we've talked about, is there something else that you were kind of thinking, or you'd like to say, or something we haven't touched on that you'd like to mention?
We've gone over quite a wide range, haven't we?
Yeah.
So that's wonderful. I think, that last point about how the inflammation
Mm,
is impairing prefrontal, um, and recognizing that, and that's, um, I think medicine right now kind of just, oh, you have long COVID, you know, see if you can stick it out. That we're really needing treatments.
mm,
Guanfacine seems to help, but I think we need many more directive treatments. We're exploring that right now, and I think that could make a big difference, so that when people get ill, they really have a chance to fully recover. Or when people have been facing adversity and been through a lot of stressors, they really can rejuvenate. We need to be able to do that for people.
yeah. Yes, that seems like a good place to end. Yeah, I don't know. I think hopefully it came through, Amy, but yeah, Mac and I were such fans of your work. And I think, when you talked about Pat Goldman Rakesh being such a big influence on you, and especially someone as an influence on women in neuroscience, I remember there's one of your papers, I think it's about the mechanism of guanfacine. It's just a review paper. But I think in the introduction, you say something like, a lot of the studies in this field were made by female neuroscientists. So I'm going to highlight their names here just as an inspiration for women and girls in science. And I just love that because I don't know, it's one of these things. It's sort of it's hard to sometimes get the tone right of how to do that. And I don't know, I just thought that was really wonderful. And I know, yeah, when, my first PhD student who's now finished, she she loves you and your work. So I mean, when she read that, she just, I don't know, found it such a kind of uplifting thing. So I think that's not only leaving a cool science legacy, yeah, a very cool legacy in that regard as well.
Thank you. Because I think, yes, now more than ever, we're getting signals again that no, we shouldn't be doing this as women. And it's
Ludicrous.
so important to have role models and to know you can thrive through a lot of adversity. I'm needing to tell myself that right now,
Yeah, yeah,
given NIH funding, and so that we can believe in ourselves. Yeah,
yeah.
and keep our prefrontals online.
Here, here.
Well, thank you so much. This has been such a great chat. I feel like we could keep chatting for hours. It's been really wonderful. Thank you for giving your time. Really appreciate it.
Total pleasure.
Yeah.
Take care.
Thanks,
Thanks
Amy. That
so
was
much.
amazing.
Bye,
Bye.
Amy. Thank you.